Hello students, a very good afternoon. So this session will be having a question and answer discussion on the test we had in the morning. Okay. So before we head on to our session, I would like to launch a technical check. Please let me know if my audio and video is clear. Okay, perfect. YouTube students, you can also put your comments on your chat box if you have any technical issues with audio or video. Okay. Please give me a confirmation if my audio and video is clear. The poll should be on your screen. Yes, perfect. Thank you very much. So let's head on with our session. And you already answered these questions. So I would like to ask, how was the paper? Did you feel it difficult, medium, or easy? Yeah. So moderate. Yeah, that's a really legitimate answer because some of the questions were very applicative, right? It's all right. So we have some of the questions which are like quite centered to NEET examination, and many of the questions were given in the test that will help you to build a better exemplar and better knowledge about some of the concepts. Okay, so we can go through that. So after this session, if you have any doubts with any of the specific questions, you can just post your question number. So if we have time, we will address that questions. Okay, thank you. So let's get on to our first question of the day. Yeah, the number of mitochondria in the cell depends on the number of chloroplasts in the cell, shape of the cell, number of chromosomes in the cell, and physiological activity of the cell. Okay, so here we have a poll. Let's take, hold on 1 second, people. Sorry. I'll just launch the poll for you. So you can answer through that. Yeah, the polls are live. So give me your answer. The number of mitochondria in the cell depends on. You should not be taking more than 30 seconds, right? So it's a very direct question. Students, what did you answer in today's exam? Is it number of chloroplasts in the cell, or is it shape of the cell, or number of chromosomes in the cell, or physiological activity of the cell, right? So these are your options. So what did you answer? We are 55 seconds up, almost a minute. So let's end the poll. Okay. So we have been learning about mitochondria for a very long time. It's called as a powerhouse of the cell. Obviously, each of the cell that is in our body, it totally depends upon, like it completely varies, like how much energy they utilize. For example, I will give you 3 examples. One, you take the cardiac cells. Second one, you take the, skeletal muscle cells. Okay, we have the skeletal muscles. In the skeletal muscles also, we have the sarcomere, right? So in the muscle fiber, just to condense it, muscle fiber, cardiac muscles, and the sperms. So these are the cells which require high amounts of energy. Okay. So when they require high energy amounts, why do they require high amounts of energy? Is because, excuse me, is because of their physiological activities. If you take an example of a skeletal muscle, right? The skeletal muscle fibers require tremendous amount of ATP to keep the muscle contraction going on, right? So we understood in muscle contraction theory that the presence of ATP is very, very essential, okay, for the cells to continue on contraction and relaxation. So the beautiful example is, why do people have very, highly contracted and stiff muscles? Is because the absence of ATP, the cross bridge cannot be broken. Are you understanding? So that is why the muscles become stiffer for the dead people. So that is one of the physiological example. Second one is when you take the example of sperms. sperms have to travel a very long journey to meet the ova for the fertilization. So if you see the neck region of the sperm, almost around 250 mitochondria are present. Okay. So even the cardiac muscles, which are continuously contracting and relaxing for the beating of heart, the myocardial cells. So even they require tremendous amount of energy. So the number of mitochondria varies from each cell to one cell to another cell solely on the physiological activity of the cell. Okay, so if you have written option D, then it is right. Okay, so it does not depend on number of chloroplast, obviously, and the shape of the cell, no, and the number of chromosomes, it doesn't determine basically. So that is why option D is very, very relevant. Okay, next one. In some bacteria synthesize their food by energy released during conversion of NH3 to NO, NO. So these are called heterotroph, chemoautotroph, photoautotroph, ammonifying bacteria. Okay, so let's relaunch the poll. Let's see what you have the answer here. Okay. Yes. Autotroph. Okay. Come on, students. Others, give your answer. What did you write in examination? Let's go. Let's go. I want to see how the paper went. Others, give your answers too. Participate actively so that we can make this session very interesting. It's all right if it is a wrong answer. Anyways, we're going to give the explanation for why it is wrong and why it is right. So you can go ahead and answer. No problem. Yes. Yeah. All right. So we are 40 seconds up. So let me end the poll and let's see what is the right option. Okay, people. So you might have heard about cyanobacteria, right? So cyanobacteria's role is to make the availability of, nitrogen. It converts NH3 to NO. So if you see the cyanobacteria, it is a photoautotroph. Okay, it uses sunlight for the convert. See, the energy is basically what they will do is they will derive the energy through photosynthesis only. Okay, they're using water for the photolysis. But what they do is by the process, they also convert NH3 into NO. Okay, so they're asking about food. Okay, since they synthesize the food. So these are called as what? They are called as photoautotrophs, people. It's not ammonifying bacteria. Okay, in ammonifying bacteria, what happens? The nitrogen, okay, in the air is converted to ammonia and it is made available for the plants. It's one of the process. But if you see here, NH3 is converted into NO, so that happens in cyanobacteria as a major example. So cyanobacteria is a photoautotroph. Okay, so you can use this as an example, right? So we use cyanobacteria as also a biofertilizer sometimes. That is because it is making the nitrogen available as an available form for the plants. So by converting NH3 into NO. All right, people. So the answer is photoautotroph. It's all right. It's not a heterotroph and it is not a chemoautotroph because chemoautotrophs are usually depending upon H2S or HNO3. So they use this things and they break down for the electrons so that they can synthesize their energy. Okay. Next question. What will be the percentage of dihybrid plants? Okay, sorry. What is the percentage of hybrid plants in a generation of Mendel's monohybrid cross? Okay. So we have this question and our options are 12.5, 50%, 6.25%, and then we have 87.5%. Okay. So I will launch the poll again, people. Nice, you're participating. I appreciate it. Let's go again. Okay. So tell me, what did you tick? What is the percentage of hybrid plants in a generation of Mendel's monohybrid cross? Okay. Nice. Yes. So nice, people. Come on, others, please answer. Make it fast. YouTube students, you can also text in the chat box. I'm following you up so that you're not missed out. So you can text your answers in the chat box. All right. So let's see if you have got the right answer and our time is up. So I'm going to end the poll. See people, we have a monohybrid cross, and after the monohybrid cross, what is monohybrid cross? We have capital T, and we cross it with small t and small t, which are homozygous plants, and they are parent plants, and we get a first filial generation where all the plants are capital T and small t, right? So while self-crossing capital T, small t into capital T and small t, self-cross, okay, specifically. So in a F2 generation, what do we get? We get capital T, and then we get capital T, small t, and then we have capital T, small t, and we have small t and small t. So out of the 100% of the plant, we get 25% of, okay, monozygous dominant plant, and then we have monozygous recessive plant, that is dwarfness, and then both of the plants express the same phenotypic traits, that is both capital T and small t are tall. But when you see the hybrid characteristics are depending upon their genotype, so it is also 25%. So this both should be 50%. Okay, so the answer is 50%, and the one that answered 50%, you're absolutely right. Okay, so that is 50%. All right, let's go for the next one. Which of the following is not genetically modified plant? BT cotton, flavor saver tomato, okay, pusa, and then swarnium, pusa swarnium, and then we have golden rice. All right, people. Again, let's launch the poll again, and I want to see how did you analyze this question. Yeah, good. All right, nice. Okay. Let's go, people. Yes, nice. We are getting some good answers, good participation. We have one more person. Okay, two more person. Come on. Let's answer this question. Okay, chalo. Can we see the right option? Okay, so anyway, most of you have answered this. I'm going to end the poll now. Okay, so don't make it so complicated. Okay, see people, some of you have written golden rice. Okay, so why not golden rice? Golden rice is a biofortified rice, which is a GMO, which is an example for genetically modified rice. What is golden rice? Golden rice is the one which is enriched with vitamin A. Are you understanding? So the people who are having night blindness, okay, to improve their eyesight, usually we don't get enough vitamin A by consumption of milk. So many of these poor people, they go for rice. Okay, so the enrichment of vitamin A, okay, the gene that is transferred into these rice plants where the vitamin A is produced. Okay, so it has to be genetically modified. It's not a hybrid plant. All right. So if you have written BT cotton is a classical example for genetic modification where we have used cry protein, okay, or cry genes for preventing the bollworm infection or bollworm pest with respect to BT cotton, right? It is Bacillus thuringiensis cotton, where we use BT gene, that is a cry gene, right? We use cry gene for this. So this is also a GMO. So when it comes to flavor saver tomato, so we usually back in the days, you remember that tomatoes, I don't know about you, but back in our days, tomatoes would have a very, low shelf life. So usually they would become softer and they would become, spoiled very soon within like 3-4 days or something like that. So what happens is ripening of the tomato depends upon a gene. Okay. So now what happens is silencing of this gene, a partial silencing of this gene is done so that a variety of flavors saver tomato is obtained by genetic modification where the pigmentation, the reddish color of the tomato, the slow ripening of the pulp and the hardness, we can pack tomatoes. You can see if you hold the tomatoes now, they're stiff and tough, right? So nice rhyming. So that's why flavor saver tomato is another example for genetically modified plant. When it comes to Pusa swarnium, usually what happens, there is a disease called as white rust. So in many of the plants that are relevant to Brassicaceae, so these Brassicaceae plants, what they did was they hybridized it, okay, to get across this white rust disease. So the Savernium is belonging to Brassicaceae. So that is Pusa savernium is a type of a hybrid that is resistant to one of the fungal diseases. So it is not a GMO, it is a hybrid plant. Rest of all of them are genetically, rest of all of them are genetically modified plants. Okay, people. So in your textbook, BT cotton and then you have golden rice as the classical examples for genetic modifications. Okay. In biofortification, we come across golden rice, and BT cotton, we come across, applications of biotechnology. Okay. And even flavor saver tomato, it's mentioned in your textbook. Whereas this one is an odd one out. So you can just go for this odd one out. Okay. You can take a chance on odd one out. All right. So next question. Which of the following plants show a bewildering diversity of floral pattern? So we have the options fig, yucca, orchid, and then we have rafflesia. Okay. So I'm going to relaunch the poll. So let's see if you have got the right option. Yes. Excellent. That was very, very fast. Nice. Good people. Let's go others. Let's see what you have written. If you're given right answers all of a sudden, then I feel very happy. That reduces my explanation also. And if you give the wrong answer, it encourages me to explain better, right? So it's both ways win-win situation. Let's go, people. Let's answer. Yes. So our time is up. I'm going to keep it to 40 seconds because it is so easy question, right? So when we talk about the biodiversity in living organisms, right, or the varieties of organisms that we have, when we took the examples of rice, around 50,000 varieties, when we take mangoes, around 1,000 varieties of mangoes, or when we take about orchids, we have around 25,000 varieties of orchid, people. Okay, 25,000 varieties of orchid we have in our ecosystems, which is showing a very, very huge amount of diversity when it comes to one species, right? So many subspecies are present when it comes to orchid. That is why orchid is one of the greatest examples for, bewildering diversity in plants. So in insects, we go for, ants. Ants have ants has tremendous amount of diversity. And more than ants, we have beetles, right? So we have around 3 lakh varieties of beetles. That is tremendously high. So any one species which is showing a wide range of variations that we call as bewildering diversity. Okay, so bewildering is an exaggeration term that is used for huge amount of diversity. Okay, don't get confused with the term. So that is orchids. All right, so let's go for the next question. So we have the two statements given. One is assertion, other one is the reason. So the assertion is, a father will never pass the gene for hemophilia to his sons. And the second, and the reason for this is, hemophilia is an autosomal linked recessive trait. In the light of the above statement, choose the correct options given below. Our option A is not correct, but R is correct. Okay, assertion and reason are correct, but reason is the right explanation of assertion. Both assertion and reason are correct, but reason is not the correct explanation of assertion. A is correct, but R is not correct. Okay, so let's go again. All right, let's go again. This is your 12th syllabus. So you know the characteristics of hemophilia, and we should be able to answer this very, very easily. Okay, let's take a minute for this question so that you will get to think. Anyway, you have answered before, but still we'll take time on this. Yes. Okay, we have our answers pouring up. Okay, so let's see if you both are right. Okay, others, please give your answers ASAP as soon as possible. Right, I'll give you 10 more seconds. Okay, so can we go solve this question? So let me end the poll first, people. So we know hemophilia, right? Hemophilia is also called as the Royal's disease, or it is a clotting disorder. If you see the clotting mechanism, right? Clotting mechanism or coagulation of blood, what we call it as, which is a very elaborate process. We have something called as clotting factors. So if you see, we have around 13 clotting factors, which are a cascade of proteins that are responsible for, a successful coagulation mechanism. So in these 13 factors, we have from factor 1 to factor 13. All right. So there are specific factors, something like factor 8, factor 9, and factor 11. So these factors 8, 9, 11 are called as anti-hemolytic factors. Okay, what are they called as? Okay, I'll write it as anti-hemolytic factor. Okay, people. So these are called as anti-hemolytic factors. Now, all these factors are very, very important to complete the coagulation mechanism. If I ask you, what is coagulation mechanism, if you will immediately tell that it is a cascade reaction. What is a cascade reaction? We need all the 13 factors to be present, and all the 13 factors have to exist and perform very functionally to have a successful coagulation. Any one factor missing, that would lead to unsuccessful coagulation or unsuccessful clotting of blood. Are you understanding? So this anti-hemolytic factors, which are factor 8, 9, 11, are present on which chromosome? It is present on the X chromosome, correct, right? So we have two types of chromosomes. One is autosomes, other one is sex chromosomes, people, right? Sex chromosomes are XX and XY, and autosomes are the rest of the chromosomes that we have already known very well and very clearly, right? So let us see what is happening here. So when we talk about this X chromosome, so they are present on the X chromosome, I told you. But what kind of an allele is this? It is a recessive allele. So hemophilia is a sex-linked recessive disorder. If you know this statement, you can very well solve this question. And second point, what you have to remember is the gene for hemophilia is present on the X chromosome, people. So now what we have? We have a mother. Let's take that we have a mother. And then we have a father. And let's say that our father has a defective gene, or let us say that he is hemophilic. So a hemophilic father would have his defective gene on the X chromosome. So when they marry, okay, when they marry, so let us say that a man who is hemophilic marries a female who is healthy. Are you understanding? Because that is according to our question. Okay, see. So if he has to pass the gene, who gets the gene? This X will go with this X, and the same X will go with this Y, and the X will go with this X, and this X will go with this Y. So what happens, people? We have X, X dash, right? And then we have XY, and then what do we have? We have X dash X, and then again we have normal XY. Since father do not have this hemophilic gene on the Y chromosome, so the son can never have hemophilia coming from the father, but he can have hemophilia when the mother is a carrier of the disease. Are you understanding? If a mother is a carrier, let us say that one of the chromosome is defective, correct, right? It is a recessive disorder. So mother becomes a carrier when one of the chromosome is defective and the other chromosome is healthy. Now, if she marries a normal man, then if this defective chromosome goes with Y, then what do we have? We have X dash Y, then the boy becomes hemophilic. Correct now, people? So the condition, right? The condition for hemophilia, when a man is hemophilic, he can never pass a gene to his son. So that is our first one, okay? Now the second statement, when we look at the second statement, hemophilia is an autosomal linked recessive trait. Now I told you it is a sex linked recessive trait, and the gene for hemophilia is present on the X chromosome. Are you understanding, people? So now what should be our answer, people? So is the assertion right? Yes. Is the reason right? No. So A is correct and the R is not correct. So our option D is the correct option that you have to choose. Did you understand this, people? Is it clear? Yeah, I hope so. Just put on the message as I move on to the next question. Okay, let me check. Yes, perfect. So let's go to the next question, people. Nice. Yeah. So we have an assertion and we have a reason here. Again, okay. The reason is genetic code is nearly universal. Okay. Reason is it is same for all organisms except for mitochondria and some of the protozoans. Choose the correct answer. Assertion is correct while reason is incorrect. Assertion is incorrect while reason is correct. Both assertion and reason are correct, but reason is the correct explanation of assertion. Okay. both assertion and reason are correct, but R is not the correct explanation of A. So let's go and launch the poll. And I'm very curious if you get the right answer. Let's see. Here is your poll. I think it should be live. Yes, it's live. I really, really need you to participate actively. Yeah, sorry, people, there was a network issue. I'm very, very sorry about it. Okay, so you have answered. Okay, let's continue. So where are we? Here, right? So let's go back to our question. Where are we? So 1 second, people, 1 second. Yeah. And I think this is the correct one, correct question. Yeah, perfect. Okay. So I hope you can hear me, and sorry for the network error. Okay. So let's go ahead. Okay, you have answered anyways. So let me end the poll. We have taken enough time, a lot of time actually. So let's go. So if you see, some of you have answered that both assertion and reason are correct, and reason is the correct explanation of assertion. Absolutely right. Okay, option D is absolutely right. So let's see why it is right. Genetic code is universal. That's what our textbook says, correct, right, people? But we have never heard about this term called as nearly universal. Why do we accept this now is because in 1986, scientists came across some of the codons in mitochondria and protozoa, right? So we know that we have WA, WAG, and UGA. Okay, UGA. I remember like this for the stop codons because it makes me easy. UAA, UAG, and UGA, it is known that these codons are supposed to be stop codons, correct, right? They do not code for any amino acids. Whereas if you observe mitochondria and some of the protozoans, in 1986, it was discovered that this UAA, okay, and UAG, they code for glutamic acid, okay, which is very, very rare. So that is why we use a term called as nearly universal, not absolutely universal. So that is why genetic code is nearly universal. Why nearly universal is because in some of the mitochondria and the protozoans, the stop codons really code for amino acid. So that is why our option D is correct. Okay. So I hope you understood this, because when we talk about universal, that means all genetic codes should behave the same way in all the organisms, from bacteria to, the complex eukaryotes or chordates. Is behaving some way and protozoans are behaving some way. We cannot use genetic code is universal. We go for nearly universal. If people have got the wrong option, I hope you have got the right explanation. Okay. So these are stop codons. They do not code for any amino acids. But in these two cases, they code for amino acids. That's why we use a term called as nearly universal. All right. So we have another question. Okay. Given below are the statements. Statement 1. Mutation is a process by which genetic variations are created through changes in the bare sequences within the gene. Next one. This is actually base sequences. Okay. Next one. Statement 2 is conventional breeding is often constrained by the availability of limited number of disease resistant genes. Both the statements are false. Statement 1 is true. Statement 2 is false. Statement 1 is false, but statement 2 is true. Both the statements are true. Okay. So let's launch the poll. Okay. It should be very, very easy if you know the concepts. Is the poll live, people? No. Yeah. It's alive now. stop the explanation. Yeah, I know. Network errors again. Sorry, people. I'm really sorry about it. Okay, it's unavoidable sometimes. It happens. Okay, so let's go. Yeah, you have answered your questions absolutely right. So I will give you an explanation for this right away. Okay, so if you see the options that we have, okay, give me a second. Yeah, let me end this. Yeah, if you see the options that we have, mutations is what sudden changes in the gene. Okay, is called as mutations, and it can occur through various means. Okay, so which basically something that causes mutations are called as mutagens. Okay, so whenever there is a change in base pairs, it leads to phenotypic and genotypic expressions completely become completely changes, and you can clearly, it's very very well visible that mutations lead to phenotypic changes, right? So mutations are not always bad, because mutation and recombination are two important factors that has created variation, and that has resulted in a tremendous amount of evolution, okay, in our biosphere. So that is why mutation leads to genetic variations, right? So whenever there is a change in base pairs from a regular gene sequence. Now, conventional breeding is often constrained by the availability of limited number of disease-resistant genes. This is correct. Why we are not able to produce more hybrid crops or more hybrid plants which can resist most of the diseases is because we don't find the gene for many of the diseases which the plants have not developed any resistance to. So many of the diseases are not cured. We have to still go for the conventional methods of using some of the fungicides or some of the pesticides, etc. So that is why this particular plant breeding process is completely limited, because on based on the availability of the genes that can help us to beat this problem. If we had enough genes to resist these diseases, we would not require GMOs. We would go for better hybridized crops only. Okay, so that is why both the statements are correct. And nice, you have chosen the correct answer. I appreciate it. Okay, so let's go for the next question. Variations in the variations in the intensity of temperature and variations in precipitation account for formation of various biomes. Next one is regional and local variations, okay, within each biome lead to the formation of a wide variety of habitats. Okay, so let me relaunch the poll and see what is your right option. Okay. Nice. Nice, people. So let's go, people. Answer. You're giving right answers. That is so nice within 15 seconds, That's nice. Okay. So after a lot of time. So let's go for the explanation of this question. So statement one, intensity and the duration of temperature and precipitation always leads to various biomes, right? We can see the tropical forest, subtropical forest, and you can see alpine forest, and you can see the higher we go, we don't find any of this biomes, right? It will become arid as we move up. And then you can see in the desert, there is no precipitation and higher temperature. We don't get usually much of the plant life. So the statement one is correct. It leads to various biomes, right? We have nice ecosystems based on the precipitation and temperature variations that has accounted for these amazing biodiversities or ecosystems. When it comes to the second option, regional and local variations within each biome lead to the formation of wide variety of habitats. So within, you can see, like, for example, even though we have tropical area, for example, we have a tropical country, India. If you see West Bengal, we have a mangrove forest, right? If you see the if you see the Western Ghats, we have the tropical forests. So what we can see is, even though there is regional variations, if you say state level or district level, you can see that within the same district, you can see arid region, and we can also see highly vegetative regions. So that is all happening because of the the local variations that has led to small habitats, and it has resulted in wide variety of formation of wide variety of habitats. So local and non-local variations are very much for creating new biomes and new habitats. So that is why both a statement A and statement B are correct. So if you've chosen option D, then you're right. Okay. So next one. Read the following statements. During succession, some species colonize an area and their populations become numerous. Orchid shows a bewildering diversity of floral patterns, many of which has evolved to attract right pollen insect. Okay, in both parasitism and predation, only one species benefits, and the interaction is determinantal to other species. Kangaroo in Australia is an example for keystone species. So how many of the statements are correct? 1, 2, 3, and 4. Okay, so let's go and launch the polls again. Make it quick, people. Okay, is the polls live? Not really. Okay. Yeah, now the poll should be live. Yes. Nice. Good, people. Good. Okay. Can we see if your answer is right? I'm going to end the poll now. Don't mind for the shortage of time. Okay. So we need right answers. So let us go for the statement 1. During succession, some species colonize an area and their populations become numerous. Absolutely right. And first organisms to come there, they have enormous amount of resources. So when they have enormous amount of resources like food, shelter, and no predation, their species are uncontrolled. So that is why their numbers become numerous. Correct, right, people? So you see, when you don't have a competition, when you don't have anybody who is coming for who is coming for your food, then you do not have to care. You just reproduce and increase your number. So the first one is right. Second option, orchids show bewildering diversity of floral patterns, many of which has evolved to attract the right pollinator insects. Okay, so go for this option again. Okay, this option is right in one way and is wrong in the other way. It's not to attract the right pollen. Okay, the orchids have different different variations and different kinds of diversity based on where they are and what kind of an environment they live in, what kind of a pollinators they have. Okay, they usually attract, they usually attract wasps, but it is not exactly that they have designed for the attraction of the insects. So the diversity is not completely depending upon this. Okay, next one. In both parasitism and predation, only one species benefits. Absolutely right. And the interaction is determinantal to other species. Correct, no, people? Much determined. One species is benefited, other species is actually determinantal. So for example, if you have any parasitic infections, the worms live happily, but we are suffering the consequences of the worms. So that is when becomes determinantal to us. So the option 3 is right. Kangaroo in the Australia is an example for keystone species. So keystone species are one of the very important organisms in an ecosystem. And kangaroo is one of the keystone species because it controls a wide range of ecosystems when it comes to Australian ecosystem. So even 4 is right. So since 1 is wrong, so we go for option C. All right. So next question. If substance S is transformed into product P with the help, okay, give me a second. Yes, of course, B or okay, so they have given that the bewildering diversity exists in orchids, okay, and it is not exactly for attracting of the bees. There are so many reasons why the orchid Orchids have wide variety of diversity and not based on the flower types. It is based on, different species, different colors, and different ways that by which they can grow. Are you following me? So they have multiple reasons, not exactly to attract one specific pollinator. So they even attract, even the ants help in pollination. Many of the other bees also help in pollination. So they don't stick to one specific pollinator. Yes, they do modify their flowers to attract, the easy, whichever the bees that are high in number in that particular ecosystem, but not for one specific species alone. Okay, it goes on and modifying so that it provides, it increases its chances, but not exactly for one particular pollinator. All right. So if you're not satisfied with it, okay, I can send you the explanation by the end of the class and I will put it in the group. Is that fine? So just put your suggestions. Yeah, 1 second. Okay, fine. So if you have a substrate S and it is transformed into product P with the help of an enzyme E that has been induced from a wild allele A, after mutation A changes into modified forms as follows. So here we have unmodified, and then we have a wild allele A, it goes under mutation. So these are the different mutation, and it produces normal enzyme even after mutation. Are you understanding, people? So after considering the above findings, which of the allele pairs will produce recessive phenotype? Are you understanding? Which of the following alleles will produce recessive phenotypes? So we have allele A, which produces normal modified enzyme, allele alpha that produces less efficient enzyme, and beta allele that produces non-functional enzyme, and gamma allele produces no enzyme at all. Okay, so let's see what you have written. Okay, so let's go. So the polls are live, people. Make it fast. Should I move this? Yeah. Yeah. Perfect. Perfect. So we have any other answers, people? I will end the poll. Okay, let's go for explanation. That will help everybody. So see, people, we have an unmodified allele. So this particular wild allele went under a mutation, but even though after it goes under a mutation, it produces a normal enzyme. So we have, if it produces a normal enzyme, then we have to find the recessive allele. Look at this. If an allele A is present, it is producing normal modified enzymes. Okay, so that means it can be dominant, right? It is able to produce when two alleles come together, it is still able to produce, a normal modified enzyme. All right. So another allele, if normal allele goes with another allele, let us say that its alpha allele is going with a wild allele. So it produces less efficient enzyme. The enzyme is produced, but the enzymatic activity might not be 100%, even the 40% efficiency. That means something is happening, right? So that is also not recessive character that we can consider. But when it comes to beta allele and gamma allele, okay, it is producing normal enzyme, isn't it, people? So if you see beta allele and gamma alleles, they do not produce any enzyme at all. It's non-functional enzyme or no enzyme at all. So if normal allele goes with non-functional enzyme, again we are getting normal allele, normal enzyme. If a normal allele goes with no enzyme at all, again we are getting normal allele. That means we have beta and gamma should be recessive so that this wild allele is able to produce a normal enzyme. Whereas in case of A allele and alpha allele, when it goes with them, even though it is mutated and it produces modified enzyme and we are getting normal enzyme, and even though if it is mutated and we are getting less efficient enzyme, that does not affect the phenotype of an organism. When a phenotype of an organism is not changed, the first two alleles, okay, the first two alleles are not recessive, whereas these two alleles are recessive. So we go for beta and gamma are the recessive phenotypes. Okay. So next one. In which of the following genus name, its characters and class phylum are correctly matched? Okay, we have genus name Ascariasis. Okay, Ascaris, and its body is segmented. They are saying male and females are distinct. That means we can differentiate between them. And the phylum is Annelida. Next one, the genus. Okay, we have frog, and then they have a tympanum that represents the ear. Fertilization is external, and the phylum is, you have amphibia. And then we have pteropus, and the skin possesses hair, and they're oviparous, and they are mammalia. And then we have Aurelia, then they have nidoblasts, organ level of organization. Okay, and then we have phylum Coelenterata. They belong to phylum Coelenterata. Tell me, people, which is rightly matched. Make it quick. Okay, let's make it in 10 seconds. Okay, it should be easy, huh? Which one is correctly matched? The poll should be live. Okay, come again, make it. Come on, people, answer, so that I will understand where you are lacking understanding, so that we can solve this, right? Okay, fine. We have another option. Yes, nice, nice. Okay, chalo. I will end the poll now. So let's see if you have got the right option. Okay, people. So Ascaris, is the body segmented? No, the body is unsegmented, right? Yeah, males and females can be distinct because the females are longer than males. That is very easy characteristics to pick up. And they belong to Annelida. That is also correct. But body segmentation is unsegmented. They are unsegmented. When it comes to genus, frog, it's so easy. Odd one out, people. Yes, do they have an external ear as a tympanum? Yes, they do. Correct, right? That is one of the first things that we learn when it comes to amphibia. They show the presence of external ear in the by having a tympanum. Now, is the fertilization external? Yes. When you go in the rainy season, you can see that small bubble-like things with a black dot inside, right? So those are nothing but frog eggs. The frog eggs are not produced, within the parent body. So the gametes are released in the local ecosystem like water and ponds or, stagnant water. So the fertilization happens externally. Okay, that is also correct. And the phylum Amphibia, absolutely right. So our option B is correct. If you choose an option B, you're right. Next one, when it comes to pteropus are nothing but bats. Okay, they are large bats. So usually the skin possesses hair. This is also correct. Are they oviparous? No, they are viviparous. Okay, and they belong to mammalia. Okay, so this option is wrong because of the oviparity. It is viviparous. Aurelia, the jellyfishes, okay, which are coelentrates, correct. They have nidoblasts, correct. Are they organ level of organization? No, they are tissue level of organization. So our option B is correct. All right, people. So next we have another question. So nitrogenous wastes, water and ions, okay, accumulate in an animal body either by A or the other means like B. So we have our option metabolic activity as A and excess secretion, indigestion and excess ingestion, metabolic activity and excess ingestion, metabolic activity and excess egestion. Okay, so let's go and answer this question as quickly as possible. It should be so easy for you. Okay, it's a very simple question, people. Yes. Fine. So nice. Good people. Okay. Chalo. Let's go for your options. So if you see nitrogenous waste, basically when we have metabolism and when we are breaking down proteins and modifying proteins, obviously we produce nitrogenous wastes, right? And water and ions also accumulate by the amount of food we take, right? It depends only on the type of food we take. So if you're taking a lot of water and if our fluids contain a lot of electrolytes and if our food contains a lot of protein rich food, obviously these three things come into play. So they accumulate in the animal body. Okay, and the other means like, so when do they accumulate? First one, I told you, it depends on the food. That means you are taking the food from outside to inside. That means you have to take it has to be metabolized, correct, right? So you take the food and then you metabolize, correct, right? So what is that? Metabolism is called as metabolic activity, breaking down. So if you have a metabolic activity and then you have excess ingestion, I told you, if you take protein rich food, you have nitrogenous waste. If you take a lot of water, then again it is called as an ingestion. If your fluids are containing a lot of electrolytes, again it is called as an ingestion. So this happens when the metabolic activity is due to metabolic activity, and then the second one is excess ingestion. Okay, it depends on what you eat, right? So based on what you eat, certain molecules accumulate in your body. Okay, people. Next one. Deficiency of which plasma protein will lead to the movement of water from capillaries to the tissues. So our option is creatinine, okay, fibrinogen, albumin, and gamma globulins. Okay, gamma globulins. So this should be very interesting question. Okay, let's see what you have answered. Good people. Nice, nice. So we have our participants answer. I hope I can go for the explanation. Okay. So let's see. If you take the blood plasma majoritarily contains 90% of water, whereas 7% of the plasma contains proteins. In the 70%, in the 7% of the protein that is present in the plasma, almost 60% of the protein is albumin. Okay. And then we have lesser percent of fibrinogen and gamma globulin. People, so fibrinogen is responsible for clotting mechanism, whereas gamma globulin is responsible for immunity. Okay, so they're responsible in immune mechanism. Whereas when it comes to albumin, so what are we talking about? When we see our circulatory system, we know that our blood cells cannot seep through our capillaries because we have a closed circulatory system. That means our blood vessels are closed. Only the blood cells are within the tube or within the capillaries. What goes out of the capillaries is your plasma. So one of the proteins that goes out is the globulins, albumins and some of the gamma globulins. I told you, gamma globulins are responsible for what? It is responsible for immune reactions, whereas albumin is very, very responsible for movement of water. Okay, because it is high in number and it can easily escape the capillaries junctions. Okay, so that is why the answer is albumin. Okay, people, so that is the right option. So we have another question. Myelin sheath increases the rate of conduction of nerve impulse, that is the assertion. Reason, in myelinated neurons, depolarization do not occur at the nodes of Ranvier. So tell me if the assertion is right or reason is the correct explanation of assertion, or if assertion is wrong and the reason is right. Okay, so you have your options. Okay, sorry, yeah. So we have our options, people. Yeah, sorry. So options, what you have chosen, give me a second, it's misbehaving. Okay, fine. So can we end this? Hello, let's see what the right option. Presence of myelinated sheath increases the rate of nerve impulses. Absolutely right. So in myelinated neurons, depolarization do not occur at the nodes of Ranvier. Is it true, people? Is it happening like that? So, or is there the jumping of electrical impulses happen because of the nodes of Ranvier? So one of the depolarization characteristics of where there is no myelin is depending upon the nodes of Ranvier. Okay, nodes of Ranvier are very important because there is where the depolarization, actually is amplified throughout the length of the axon. So both assertion and reason, okay, so your assertion is also true, okay, and the reason is not the correct explanation of the assertion. Okay, it's not the correct explanation of the assertion. Sorry, people. Nodes of Ranvier, we don't have depolarization, but both the reason, assertion and reason are true, but reason is not the correct explanation of assertion. So you have chosen option B, and option B is correct. Okay, sorry, so there was a mismatch. So your option B is correct. Is it clear, people? So myelin sheath increases the rate of nerve impulse, and the second one, in myelinated neurons, depolarization do not occur at the nodes of Ranvier. Both are correct, but it's not the right explanation of assertion. So we have another question. Thymus gland secretes steroid hormones called as thymosin, and thymosin plays an important role in cell-mediated and humoral immunity. So both statement A and B are correct. Both statements A and B are wrong. Only statement A is correct, or only statement B is correct. Make it fast, people. Let's see what's the right option. Yeah. So chalo. So some of you have mixed options, and I understand the reason. You are so right that thymosin secretes, okay, sorry, thymus gland secretes thymosin. But where you fail to understand it, it's not a steroid hormone, it's a peptide hormone. Okay, steroid hormones and peptide hormones are so different in their characteristics. All right, so the steroid is wrong. Okay, people. Next one, thymosin plays an important role in cell-mediated immunity. Absolutely right. Okay, they're responsible for production of our antibodies as well as our T cells. Those stimulate the production of the T cells and the B cells. Okay, so what should be our answer? Only statement B is correct. Okay, the reason why you got mixed up is because of the steroid hormone. It's not steroid, it is peptide hormone, people. So that is your right option. I hope you understood this. Next one. The testes are situated outside the abdominal cavity within a pouch called a scrotum, and the scrotum helps in maintaining the lower body temperature, low temperature necessary for spermatogenesis. So this should be so much easier because you have already studied in your second year, right? So when it comes to male reproductive system. So what do you think, people? Yeah. Perfect. Okay, let's go for explanation. Already you have given the right option. So testes are located outside the abdominal cavity within a pouch called as scrotum. Yes. Why they're present? Because to maintain a suitable temperature that increases spermatogenesis. So if you see the testes are two sets of muscles. One is cremaster muscles and dartus muscles, we call. So this both cremaster muscle and dartus muscles, they help in ascending or descending of the testes based on the external environment. Okay, so thereby cooling or warming the testis. If it's too cold outside, it will contract and it will move the testis near to the abdomen. If it is too hot outside, it will descend down and it will make the testis cool faster compared to that of the outside environment, thereby providing a suitable temperature for the spermatogenesis. All right, so both assertion and reason are correct, and reason is the correct explanation of assertion. Your option A is absolutely right. Okay, so infected cells can survive when the virus are being replicated and released. Okay, Okay, in this statement, which cell acts as an HIV virus factory? Okay, so we have T helper cells, and we have macrophages, T killer cells, and we have T suppressors. So this is an applicative question. You have read about this already very detailedly, but all you have to do is apply a little bit of, your sense into it. Perfect. Okay, exactly. It's straight away mentioned in our textbook. I'm going to end the poll, people. You have given quick answers. That's nice. So infected cells can survive, okay, while the viruses are being replicated. Absolutely right. So that infected cell that can survive is basically macrophages. So macrophages are one cells that act as a HIV factory, right? They produce a lot of, proviruses using the macrophages. So they modify the DNA, and the DNA produces a lot of viral RNAs, and each viral RNA, becomes a new provirus, right? So macrophages are called as HIV factory. It is so well given in your textbook. Okay, macrophages act like a HIV factory. So you have chosen a right option, that is macrophages, and your option B is correct. All right, people. So we are running out of time. I hope you did well in your examination and you had a good time in this question solving sessions. Okay. Thank you very much. Thank you. Thank you, people. Okay, it was interesting. Thank you for your responses. It made the session very nice. So I will end the session now and I'll see you in the next class. Bye-bye.